this lecture, we will briefly touch on Chapter 27, Vaccines. We're not going to go into a lot of detail, so I encourage you, if you want to research anything in a little bit more depth, to please do so. For now, we're just going to kind of basically go over the pathophysiology of vaccines as far as how they work. So again, immunity is dependent on being introduced to some sort of antigen and then developing immunity to that particular antigen through some sort of artificial or natural means. Now, the concept of vaccination, or actually deliberately introducing a potentially harmful microbe into a patient, was originally met with suspicion and outrage, which is to be expected. Now, the goal of vaccination is to produce artificially acquired active immunity against a specific disease. Now, today, vaccinations against any sort of contagious disease is considered to have a very positive influence worldwide. Now, part of the success of vaccination is through what we consider to be called herd immunity, although I don't really like the way the textbook refers to this, because they refer to herd immunity as through the process of a widespread vaccination program. However, up until several years ago, herd immunity was considered just natural immunity that happens through the response of natural exposure of one person contracting an illness or infectious agent and then spreading that to someone else who also will get that illness, and then the body will develop its own natural immune response. So an example of this would have been an outbreak of chickenpox when you were small, and that, all the children that you're around all come in contact with it, and then they all develop the immunity. Now there's vaccines for chickenpox. So this is where your textbook is referring to that herd immunity by an artificial means, not a natural means. Now, the original purpose of vaccines was intended to actually stimulate an active immunity and to actually create an immune memory, so that if there is a subsequent exposure to that same microorganism, then it will stimulate an already primed immune response to actually fight against it. So traditionally, vaccines were basically a biological suspension of some sort of weakened or killed part of the pathogen, or the whole pathogen itself, so that it doesn't actually cause disease. But by exposing the patient to the dead portion of it, or the entire portion of that particular pathogen, then it would actually boost the patient's immune response and allow them to generate antibodies against that particular dead pathogen. And typically that pathogen is going to be some sort of bacteria or a virus. Now, the history of vaccination actually begins as early as 1000 BC before Christ, when the Chinese would actually take a pulverized powder that they made from a smallpox scab, and they would actually use the process of virilation, where they would take the patient and create a small scratch on the surface of the skin, and then they would lay this powder over it to actually initiate an immune response against the virus and create immunization. Now, by the 18th century, this practice was also adapted by the Europeans and Americans. And through all of these various scientific discoveries and research, it has actually led to the development of numerous types of vaccines that will produce an immune response in order to hopefully protect us against any sort of infectious disease. Now, each vaccine that is produced by different methods, so each of them actually has advantages and disadvantages. Now, most categories of vaccines are divided into just two, with inactive vaccines as well as live attenuated vaccines. Now, with your inactivated vaccines, these are produced by actually killing the pathogen in order to create an immune response. So these are most of your traditional vaccines, where they actually use something to denature and inactivate and kill that particular pathogen that they're looking to develop the vaccine against. And they usually can do this by something like heating it up or chemically treating it in order to kill it, or even use a form of radiation to actually kill it. And then from there, by initiating that weakened or dead particle into the individual, then it will actually elicit an immune response. And active vaccines are usually more stable and safer than an actual live vaccine. The dead microbes cannot mutate back to their disease-causing state, so that is the positive side of it. They usually do not require any sort of refrigeration, and they can be stored and shipped in a freeze-dried type of form. Now, one of the disadvantages that has been seen is that they can stimulate a weaker immune system response compared to the live vaccines. So therefore, some patients may not elicit that immune response like they should, so they could potentially need boosters in order to develop that full immunity. Now, the live attenuated vaccines are created by modifying a disease-producing wild virus or bacteria that has been weakened in the laboratory in order to prevent that organism from causing disease. So therefore, a live attenuated vaccine is actually the closest thing to being exposed to that particular microorganism in a natural environment. These vaccines do tend to generate a stronger cellular and antibody type of response, and they will often produce a lifelong immunity in the patient after only 1 or 2 doses. So when we talk about attenuated vaccines, it just means that they are weaker. So you have those live particles, they're just weaker. So viruses can actually be attenuated through a method of growing generations of them in cells in a hostile environment. So as they continue to grow and develop, they have to adapt to this new environment, and then they become weaker and weaker infectious agents compared to their original, how they were originally would have responded in a natural host. Now, based off of modern genetic engineering techniques, they have been able to create what they call chimeric virus. And these viruses will actually contain the genetic information and actually display the biological properties of that particular virus, but they'll just kind of display it in a different way. So gene-based vaccines were originally designed to encode for a chosen viral protein in the DNA or the messenger RNA. Now, this category is one of the newer vaccine methodologies where DNA and messenger RNA vaccines are actually able to induce both a specific humoral and cellular type of immune response and allow a high degree of adaptability to encode for a particular antigen. In terms of manufacturing, both platforms allow production of different vaccines using the same established production process and facility. Now, for your DNA vaccines, what scientists will actually do is try to actually analyze the various genes of a microbe, and then they will actually attempt to create that DNA vaccine against that particular, the gene within that particular bacteria. Now, DNA vaccines dispense with both the whole organism and components of the organism. So they're actually using genes that will code for the antigens of that organism. So it was actually discovered that when the genes for a particular microbe's antigens are actually introduced into the human body, then some of those cells in the body will actually take up that DNA. Then that DNA will actually instruct those cells to make that particular antigen molecule. So then those cells will actually start to secrete those antigens to the surface, and so those cells will display on the surface. And in that way, the patient's body will actually become a vaccine maker. So it's creating the antigens, and then the body will stimulate and make those antibodies. And so as your textbook kind of explains this whole process, it's letting you know that the DNA vaccine against a microbe will actually elicit that antibody response to those free-floating antigens that are actually secreted by the cells. And of course, that vaccine is supposed to stimulate some sort of strong cellular response against the microbe antigens that are on the cell surface. And so the textbook also states that the DNA vaccine does not cause the disease, but it does, that it does not actually contain the microbe. It just contains copies of the genes to actually make those antigens of that microbe. Now, RNA vaccines represent the most recently developed technology, and so their use in humans is less well characterized than it is for the DNA-based vaccines. Now, the actual messenger RNA molecule itself, it's actually composed of nucleotides that are linked in a unique order. And what that does is it actually conveys genetic information for the cell to produce the particular protein or antigen that is actually encoded by that messenger RNA. So when a messenger RNA is actually in a vaccine, from there, what it does is it actually is internalized by those particular cells or by cells throughout the body, and those cells will rely on that genetic template that is actually inside of that messenger RNA to actually translate that genetic information, and then those cells will actually start to produce those antigens that are encoded by that messenger RNA. And then, of course, those antigens are going to be expressed on the cell surface, and then the body will recognize them and elicit immune response. Now, there are two major types of messenger RNA that have been used in the development of vaccines for the messenger RNA vaccines, and these are non-replicating messenger RNA and then self-amplifying messenger RNA. The non-replicating messenger RNA contains the sequence of antigens of choice that are flanked by the 5' and the 3' untranslated regions, and this has to do with that whole cellular DNA molecule makeup. Now, the advantage of using the non-replicating messenger RNA vaccines compared to the self-amplifying ones are that they are rooted in simplicity of the construction, the small size of the RNA, and the absence of any sort of additional encoded proteins that could potentially induce some sort of an unintended immune response. The self-amplifying messenger RNA vaccines are most commonly based on the alpha virus genome, from which the genes are actually encoding the structural proteins that have been replaced with a particular antigen of choice. Now, despite these gene deletions, the viral RNA is replicated and transcribed by the viral RNA polymerase. Now, lower yields and increased occurrences of abortive constructions as a consequence of the large size of that particular vaccine molecule, and it poses challenge to the production of it. Now, any sort of genetic information that's actually encoded for by that self-amplifying messenger RNA vaccine will actually be amplified many, many times, which will actually result in a very high level of antigen expression from just basically a very low dose of a vaccine. Self-amplifying messenger RNA is most commonly delivered with some sort of synthetic delivery vehicles. So this technology actually requires the use of an electroporation of the genetic material inside of the cells during the manufacturing process. So what it actually does is uses electrical charges and impulses to actually force that messenger RNA inside of the cells. Now, there has been some safety concerns with this because this delivery method can actually lead to the presence of replicon and helper RNAs that could actually lead to the generation of the infectious virus itself. So back in the early 1990s, scientists at the National Institute for Health actually discovered that recombinant protein antigens from the outer shell surface of the HPV, or human papillomavirus, it can actually form particles that closely resemble the actual virus. Now, these virus-like particles are multi-protein structures that mimic the organization and conformation of authentic native viruses, but they actually lack the viral genome. Now, the virus-like particles will actually prompt an immune response that's similar to the elicited response that we have by the actual natural virus. But these virus-like proteins are non-infectious because they do not actually contain the genetic material that the virus will need in order to replicate inside of a cell. Now, the subunit vaccines, which are just like the inactivated whole cell vaccines, they do not contain any sort of live component of the particular pathogen. They differ from the inactive whole cell vaccines because they only contain the antigenic parts of the pathogen. So these parts are actually necessary in order to elicit that protective immune response. Now, this precision actually comes at a cost because an antigenic property of the various potential subunits of that pathogen actually has to be examined in detail in order to determine which particular combinations will provide a very effective immune response within the correct pathway or to initiate the correct pathway. Often you can actually elicit a response, but there's no guarantee that the immunological memory will actually be formed in the correct manner. Critical to the protective effect of the actual subunit vaccine itself, there is something that's added to them called adjuvants, and we did discuss this in another lecture. And what these will actually do is they're added into the vaccine to actually help amplify the immune response between the antigens alone, because the antigens alone are not sufficient enough to actually induce an adequate long-term immunity. So the adjuvants are actually tested for safety and continuously monitored by the CDC and the FDA, but currently aluminum and monophosphoryl A are the two adjuvants that are licensed for use within the United States. I do encourage you to look those up and just kind of do your own research about how that all works and combines together in order to help those vaccines to go inside of those cells and to elicit that immune response. And I encourage you to look those up and see what kind of role they actually play in the body. The recombinant protein vaccines will actually present an antigen to the immune system without viral particles, and it does this using a specific isolated protein of that pathogen. Now, it may bind to different antibodies than what the actual pathogen protein that it was intended to do. And of course, development of this particular subunit vaccine is done through the recombinant DNA technology. And this is a technique where the genetic material from one organism is actually inserted into a foreign cell of another organism in order to mass produce the protein encoded by the inserted genes. And so this is also known as recombinant genetic engineering. All right, so continuing on with the subunit vaccines, there is a vaccine called a conjugate vaccine. Now, what this does is it actually uses the scientific knowledge of bacteria themselves. So what we know is that bacteria have a polysaccharide type of coating, which is basically a carbohydrate of some sort with many sugar molecules that are bound together. Now, that particular polysaccharide coating can be made into a vaccine. And so what it does is it actually takes those particular proteins that are unique to that pathogen, and then you give that to the body, and then you basically initiate an immune response. And so with the conjugation part of it, what happens there is that it will actually change those immune system responses from being a T cell independent to a T cell dependent one, which will produce an increased immunogenicity for that vaccine for infants. And then, of course, when you give boosters, it helps to elicit that antibody boost. One thing to note, though, is that the antibodies that are induced by this type of polysaccharide antigen has less functional activity than those induced by protein antigens. There are a couple of diseases out there that the disease process actually comes from the toxin that's produced, not the organism itself. So there are vaccines that are toxoid vaccines, and what these do are used when a bacterial toxin is the main cause of the actual illness. And so it actually contains a harmless toxoid that stimulates the immune system to produce antibodies that will react to and block the particular toxin once it comes into the body. And so an example of this is tetanus. So vaccines can be administered in a variety of ways in order to maximize the effectiveness of that vaccine. And the site of administration is going to be dependent on the vaccine itself as well as the age and the size of the patient. Now, they can be given orally as well as through a nasal spray. And so you may be familiar with the nasal influenza vaccines. And this is just a needle-free approach in which the vaccine is sprayed into the nostrils and then it is absorbed through the nasal mucosa. Vaccines can also be injected intramuscularly, subcutaneous, or intradermal. With intramuscular administration, it goes directly into the muscle mass. And this is definitely going to be for vaccines that contain adjuvants in order to reduce the adverse effects that happen locally with the injection and those adjuvants that are inside of that vaccine. The subcutaneous is administered to the subcutaneous layer that's just above the muscle, but it's below the skin. The intradermal are administered to the topmost layer of the skin, and so this the vaccine that is administered this way is the BCG vaccine, and that's because it helps to reduce the risk of any sort of neurovascular injury. Some of the messenger RNA vaccines can be delivered directly into the skin, and that's because this is an organ that is populated with those antigen-presenting cells. And so what that does is it allows those cells to actually uptake that naked messenger RNA and then actually use it within those cells and elicit that humoral and cellular response. So again, I'm not expecting you to know a whole lot about this chapter. I just kind of wanted you familiar with vaccines as far as their delivery method and how they actually initiate that response and what is in them that actually stimulates that immune response, just so that you can understand a little bit about the technology that is kind of evolving based off of the newer science technologies.